So, dear students, welcome to the second lecture of the chapter Biotechnology, Principles and Processes. So, in the previous lecture, we stopped our discussion on cloning vectors, right? So, in this chapter, we will continue that topic as well as look into rest of the topics of this chapter and ultimately finish this chapter in this lecture only. So, so here is the figure given for a cloning vector named PBR322. So this is a plasmid. It is isolated from E. coli. So in this cloning vector, you can see a few labeled components. So what are these? First is the or origin of replication. Okay, that is the start point from where the DNA replication will begin, right? And plasmid is autonomously replicating extra chromosomal circular DNA, right? So it will replicate on its own without the help of the chromosome of the host cell, but it will need the DNA polymerase from the host cell. Okay. Other components, there are antibiotic resistance genes. So there are two genes actually. One is AMPR, which stands for amphicycline, okay, the antibiotic amphicycline resistant gene, and TET, that stands for the antibiotic tetracycline, and this gene is called tetracycline resistant gene. So if any if any bacterial cell is having this kind of antibiotic resistant gene, then that particular bacterial cell can grow easily in a media containing that particular type of antibiotic. Otherwise, the bacterial cell will not grow on the medium. Now, these kind of genes, they are called selectable markers. Why they are called selectable markers, we'll come to it in a minute. Apart from these two important components, there are cloning sites, which is another name for the recognition sequences for various expression endonucleases. Okay. So these are the various sites, the cloning sites. EcoR1. Okay, EcoR1 is the cloning site for the restriction endonuclease EcoR1. Similarly, there is HIND3, BAMH1, PVU1, PST1, HIND3. So they are distributed all over the plasmid vector. You should know that whenever this particular plasmid vector is exposed to, say, any particular type of the restriction enzyme, say when it is incubated with enzyme EcoR1, then the plasmid vector will be cut here where there is the recognition sequence for EcoR1. Now there is another gene called ROB gene. Now this gene is important for this particular plasmid to replicate. Okay, but these three anti-resistance gene and cloning sites, these three components are quite common in other kinds of cloning vectors like this. Okay, this ROB gene is specifically found in this plasmid vector. Now, ORI, as I already told, is a sequence from where the DNA replication starts. Okay, and this sequence has another role. It also controls how many number of copies will be made as a result of replication. Okay, so basically it dictates how many times the DNA replication process will be repeated, and ultimately how many number of copies of the recombinant DNA or the plasmid vector will be formed. Now, selectable markers, as I told, the antibiotic resistant genes, the amphicycline R and the tetracycline R, these are the antibiotic resistance genes. Now, these are called selectable markers. Why? Because these sequences or these regions, DNA sequences, they help in identifying and eliminating the non-transformants and selectively permits the growth of transformants. Okay, so the non-transformants means those cells which are not carrying your recombinant DNA, okay, or is not carrying your gene of interest, so the DNA fragment that you intended to introduce within the cell, but the transformants in which you have introduced your gene of interest. So using these selectable markers, these genes, you can selectively identify the transformants from the non-transformants. Now there are selectable markers which also help in identifying uh, the cells which are carrying your recombinant DNA from the cells which are not carrying your recombinant DNA. Okay, so that is also possible. You will explain this, you will understand this much better in a subsequent discussion. So let me first briefly define what is transformation. So you have learned from Griffith experiment, right? If you remember, uh, in the chapter molecular uses of inheritance. So he actually identified a transforming principle that was changing the phenotype of R strain bacteria into an S strain bacteria. So there the transformation happened. So now this transformation is routinely done as a procedure, okay, in biotechnology. So where people introduce a piece of DNA into a host cell, like this can be a bacteria. Here I have written bacteria, but it can be other types of cell as well, like plant or animal, or or it can be fungi. Okay, so any type of host cell, you can, if when you introduce a piece of DNA, so this process will be called transformation. Now, I told you that these are the selectable markers. Okay, so this amphicycline resistance gene, tetracycline resistance gene. This is kanamycin. This is another antibiotic. If this kanamycin resistant gene is present, then that particular host cell will be resistant to kanamycin antibiotic. So these are all selectable markers. Now we'll deeply understand the role of selectable markers in this discussion from now onwards. Now, E. coli normally is not resistant to antibiotics, okay, in any antibiotics. But when you transform E. coli, means when you introduce a antibiotic resistance gene, then due to the expression of this gene, the E. coli cell will become resistant to to a particular type of antibiotic. So if you introduce ampicillin resistant gene to a normal E. coli, then it will become resistant to amphicycline and easily will grow in a media containing amphicycline. Okay. Now, what are cloning sites? So, cloning sites, as I told you, these are basically the recognition sequences. Okay. These are basically recognition sequences which are unique to a particular type of restriction enzyme. So when you digest, or when you want to insert your gene of interest or foreign piece of DNA into a vector, you have to digest both your foreign DNA as well as the vector DNA with the same restriction enzyme. Okay, and where the cut will be made? The cut will be made at the cloning sites. Now, for one restriction enzyme, one recognition sequence should be present in the cloning vector. There cannot be multiple recognition sequences for the same enzyme. Okay, if it is so, then the number of fragments of the plasmid vector or any vector DNA will be, um, will become difficult for you to handle. Simplest ways, like if there is one site, one cloning site corresponding to one restriction enzyme, then it will basically open up the circular plasmid into a linear one, or it will basically, or if it is a linear DNA, it will then basically divide it into two parts. Okay, so two parts, it is easy to handle because you will then insert your foreign piece of DNA in between the two parts and ligate. So it is easy. But if it happening to be 4 5 fragments, then the sequence in which they will be ligated with each other may change the ultimate meaning of that um, vector DNA altogether. Okay, so that's why only one site, cloning site should be there, which will correspond to one restriction enzyme. Now, first, we will understand how the selectable markers of antibody resistance gene is helpful in selecting the recombinant cells from non-recombinants. By recombinant cells, I mean the cells containing your recombinant DNA. Okay, so how recombinants are different from transformants? So transformant cells are basically could be any cell which is having a foreign piece of DNA, okay, which you have introduced. Now this foreign piece of DNA doesn't necessarily have to be a recombinant DNA, okay. Whereas the recombinants will carry your recombinant DNA only where your gene of interest is ligated with a vector. That is how these two terms differ from each other. Now, as I was saying, that we will discuss how the selectable marker antibiotic resistance gene is going to help you to select recombinants from non-recombinants by a method that is called insertional inactivation. So let's consider the example of a plasmid DNA that we just saw, PBR322. And this plasmid DNA consists of two antibiotic resistance genes, that is amplicillin resistance gene and tetracycline resistance gene. Okay. Now, how these two antibiotic resistance genes are going to help? So, these two resistant genes consist of these cloning sites, which are there within their sequence. Okay. Now, suppose you decide to cut your plasmid DNA with the restriction enzyme BAMH1. So if you do that, then here the cut will be made, right? And within the cut, so the cut is made within the tetracycline resistant gene sequence, and your foreign piece of DNA will be ligated at this point, where initially there was BAMH1 recognition sequence. So here what you are doing, you're inserting your foreign piece of DNA within the tetracycline resistant gene sequence, because of which the sequence is now interrupted. It is not a complete sequence. So the sequence is not complete. It will not, give the meaning, correct meaning. It will be then in inactive state. Okay. So this process that you're doing, you're inactivating a gene by inserting a foreign piece of DNA in between its sequence or within its sequence. This is called insertional inactivation. Now, the recombinant DNA that you have formed is having active amplicillin resistance gene, whereas the tetracycline resistance gene is inactive. Okay, so after you have made this recombinant DNA, you will now introduce into a host cell. Okay, so any host cell. So here I've considered E. coli will transform one E. coli cell. Now these E. coli cells will be allowed to grow in a particular media and they will form colonies. So two types of colonies will be now made. Okay, one set of colonies will be transformants, that is carrying your um, foreign piece of DNA that you have introduced. So here you have introduced a recombinant DNA, so that is for the cell is foreign. Okay, and another group of cells will not be having this particular DNA piece. Correct. Now, among the transformants, it is possible that due to human error, some of the transformants is not having your uh, inserted foreign gene that is there in the tetracycline resistance gene sequence, and some may be having this gene. Okay, So you can similar. So because of this, you can divide the group of cells into recombinants and non-recombinants. So human errors you have to take into account, okay, while doing this kind of experiments. That is why after the steps are done, these selectable markers are helpful in selecting out the correct type of population by taking into account that some error might have been made in the process. Okay. So ultimately, your ultimate goal is to select out the cells, host cells, that is carrying your foreign piece of DNA ligated with the vector DNA, that is plasmid DNA. Okay, that is why this is encircled in green color. This is the population of cell that you are looking for. So how you will do? So first, plate the transformed E. coli cells, okay, which is having the foreign piece of DNA on a media containing amphicycline. Okay. Now let me ask you, what type of E. coli cells will grow on this particular medium? Whether the transformants will grow, the non-transformants will grow. Remember the definition of transformants and non-transformants. Transformants are those cells that is carrying your foreign piece of DNA. Now, what foreign piece of DNA you have introduced? You have introduced plasmid DNA and the ligated with the your gene of interest. Okay, but the non-transformants is not having this. So, if you have guessed that transformants will grow, you are correct. It is the transformants who is having the active sequence of amphicycline resistance gene, because of which they will grow in a media containing amphicycline. Whereas the normal E. coli cells or the non-transformants is not resistant to amphicycline, so hence they will not grow. Now you take the colonies from step 1 that have grown in your plate, okay, and plate them in another petri dish where you have a media containing the antibiotic tetracycline. Okay. So now guess what type of E. coli cells will grow in this particular medium, whether it will be the recombinants or non-recombinants. Okay. So again, recall the definition of recombinants. Here, recombinants, I mean that you have successfully ligated your gene of interest with your plasmid vector. Okay. And that's the single unit, the single now recombinant DNA will has been now taken up by the host cell successfully. Then whether non-recombinants, it might have happened that the ligation of your gene of interest within the plasmid DNA hasn't happened properly. Okay, so maybe the cells which have taken up the foreign DNA is not containing the gene of interest and is only having the plasmid DNA. So it is a possibility. So this is how recombinants are different from non-recombinants. So guess in what kind of media they will grow. Okay. So guess which cell population will grow in the media containing tetracycline. So if you have guessed the non-recombinants, you are correct, because the recombinants, the tetracycline gene is switched off or is inactivated due to insertion of the plasmid DNA, insertion of your gene of interest within the tetracycline resistance gene at the BAMH1 site. Remember, I told you just few minutes back. So in the recombinants, there is insertional inactivation of the tetracycline resistance gene. So that is why these recombinants will not show resistance to tetracycline. But the non-recombinants who are still having the tetracycline resistant gene intact without any interruption, so they will show this resistance. So this slide basically summarizes what we have discussed so far. The transformed E. coli cells with amphicycline resistance gene will grow in amphicycline containing medium. Now, among the transformed colonies, non-recombinant E. coli, where both these resistant genes are intact. They will grow in tetracycline containing media, as well as well means they will grow in tetracycline containing media as well. Okay, but the recombinants with intact ampicillin resistance gene, but the tetracycline resistance gene is inactivated. They will only grow in ampicillin resistance gene, okay, but not in tetracycline resistance gene, but will not grow in tetracycline containing medium. So up till now, I think it is clear that how these two resistant genes are acting as selectable markers to select out uh, first transformants from non-transformants, and then among transformants, the recombinants from non-recombinants. Now, limitation of this approach is that how will you ultimately then grow the recombinants, which is not able to grow in tetracycline containing media? How will you ultimately grow them, because they are the population we are looking for? So one way is to you have to isolate the plasmid DNA from all the colonies of the transformants, okay, and then analyze like which of the transformed cell actually having your gene of interest within its plasmid DNA. So this is going to be quite time taking and cumbersome process, and is going to obviously add in more and more human errors that will make your analysis difficult. So another approach could be using another type of selectable marker. So let's try with the selectable marker named beta galactosidase gene. So you have heard this gene when you were learning about lac-operon. Okay. So how this gene is helpful here? So this particular gene, also called LAC-Z gene from the lac-operon concept. So this gene actually codes for the enzyme beta galactosidase. Okay, and this enzyme actually acts on substrate lactose. Okay, if you remember, and then converts it into glucose and galactose. But here in the media, in the culture media, we are going to add a substrate which is quite similar to lactose, and it is called XII. Okay, it is a synthetic analog. It is produced synthetically and very much similar in structure to with lactose. And this substrate is actually colorless. It doesn't have a color. So when when beta galactosidase acts on the substrate XII, which is colorless initially, it produces a product which is blue in color. So the final product here will be blue in color. So if we can write LAG-Z gene, okay, makes the enzyme beta galactosidase, correct? And when this enzyme acts on XCal, which is colorless, it produces a product which is blue in color, blue colored product. So we are getting a colorogenic product. Now, how this is going to help us in selecting out the recombinants from non-recombinants? Now consider another cloning vector, PUC. Okay, so it also has ORI, and it is having antibiotic resistance gene here. It is amphicycline resistance gene, and it is having a part of the beta galactosidase gene, but this part is all you need for this reaction to happen. Okay, so this is the only part you need. So this part is there. It's called LAC-Z prime. Okay, and within LAC-Z prime there are multiple cloning sites. Basically, the recognition sequences for different types of restriction endonucleases, as well as there is a gene for the gene that codes for the repressor protein. Now, if you recall your lac-operon concept, this repressor protein is going to go and bind with the promoter of beta galactosidase gene, that is here LAC-Z prime. and going to inactivate the synthesis of this enzyme. Okay, but it will only happen in the absence of substrate. That is, in lactoperon it was lactose, but here XGAL. So when XGAL is present, the repressor protein will be inactivated. So the lag Z prime can be synthesized normally. Now for this plasmid vector, consider that you want to link a foreign piece of DNA and then make a recombinant DNA basically. So where this foreign piece of DNA will be ligated, it will be ligated here. Okay. So because of that, uh, this sequence, lag Z prime sequence, will become inactive. Okay. And the beta galactosidase gene will not be produced. Okay. So you do first step one. You first uh, plate the E. coli cells that you have transformed with this particular recombinant DNA in amphicycline resistance gene. So from this particular gene, you will get to know whether or not the cell has taken up your DNA, foreign piece of DNA or not, means recombinant DNA or not, whether it has transformed or not. So if it is transformed, then it will successfully grow in amphicycline resistance gene. So here, only the transformants will grow in amphicycline resistance gene and amphicycline resistant, amphicycline containing media. And the second step, you take these E. coli cells, transformed E. coli cells from step one, and grow them in a media containing XGAL. Remember, this is the substrate for beta galactosidase. Now guess what type of E. coli colonies you will get in this media, whether it will be recombinants or non-recombinants. So answer is you will get both types. Okay, both the recombinants, which is carrying your recombinant DNA, where the foreign piece of DNA has been successfully ligated at MCS, okay, multiple cloning site, as well as the non-recombinants, where the foreign piece of DNA is not there at the cloning sites. Both will grow. But the only difference is the recombinants will be white in color, whereas the non-recombinants will be blue in color. Why? Because the non-recombinants, there in those cells, beta galactosidase will be synthesized. Okay, and it will consume this substrate and create a blue colored product. Whereas the recombinants, where you have inserted your gene of interest within the sequence of lag Z prime, so because of which the lag Z prime gene is inactivated, so beta galactosidase gene will not be produced, or means will not be synthesized, means beta galactosidase enzyme will not be synthesized. So it will not consume the XCal substrate. So because of which all the E. coli colonies will be white. Now, through simple observation, you can means select out the white colonies and then take forward your subsequent steps of recombinant technology. Okay. Now, various type of vectors are used for cloning genes in plants and animals. So this up till now, the plasmids that we have talked about, PUC and then PBR322, they're usually used for transforming bacterial cells. But what about transforming plants or animal cells? So we'll talk about those here. So for transforming plants, people have discovered a plasmid called TI plasmid, which is found natively in a pathogen that is called Agrobacterium tumefaciens. Let me repeat, Agrobacterium tumefaciens. Now these are pathogenic means microbes, okay, and they are known to trigger the transformation of normal plant cells into tumor cells. Okay, so because the TI plasmid basically contains a DNA called tDNA, and this tDNA is actually causing transformation of normal plant cell into tumor cell. So if we remove this tDNA from the TI plasmid. And then the pathogen is disarmed, means it cannot cause tumor. Instead, what we can do, in place of tDNA, we can link our non-harming gene of interest, okay, in place of tDNA within the TI plasmid, and now allow the Agrobacterium tumefacium pathogen to infect the plant cells. Because of which the plant cells will be transformed and will show the character which we intended by introducing this gene of interest. Okay, this is what I have written, that tDNA, when expressed, can transform normal plant cells into a tumor and direct these tumors to produce chemicals required for Agrobacterium tumefaciens. Okay. Now, for transforming animals, people are using retroviruses. Okay, so retroviruses has been known to cause cancer. Okay, that is, they infect the normal animal cells as well as the retroviruses for all types of mammals, humans as well. So they can infect normal animal cell and transform into tumor cell. So here also the same logic can be applied. If we remove the cancer causing genes from the retroviruses and instead add your gene of interest, okay, which you want to express in your organism, and then allow, now you allow this retrovirus to infect, okay, your animal. So you will see that the animal's phenotype or the animal cell is showing the desired phenotype. So this is how vectors are used for plants and animals. Now, in order to introduce your recombinant DNA within a host cell, you have to first make the host cell competent. So what do I mean by competent and competent, or how you will make the host cell competent, or why there is a need to make a host cell competent? So competent means much more willing to take up something foreign. Okay. Normally cells are not that much eager to take up anything that they don't want to. We have to force, you're actually going to have to force your recombinant DNA into the host cell. So that is called making the host cell competent, so that you can force something into it. And why you need to do that? Because DNA, as you know, is a hydrophilic material. Okay, it's a hydrophilic molecule. That is, it loves water or aqueous environment. But the cell membrane, is composed of lipid bilayers. So it is hydrophobic. So it will not allow any hydrophilic molecule to enter easily. Okay, which is why you have to perform some steps to make pores at the cell membrane through which your DNA molecule can enter easily. So what are those steps? First step, you have to first treat your cells with a specific concentration of calcium ions. Okay. For example, calcium phosphate. And that too you have to do on ice. Okay. So when you do that, the cell membrane will be made permeable, or or you will artificially create pores on the cell wall of the cell membrane. After it is done, now these steps are done for bacterial cells. Okay, these steps that I'm talking about is for making a bacterial cell competent. Now the next step is that you have to then incubate your bacterial cells with your recombinant DNA on ice. You have to keep it for a few minutes. After you do that, you have to give a heat shock, means expose the mixture to high temperature, that is 42 degrees Celsius, for 60 to 120 seconds, means 2 minutes hardly. Now this heat shock will actually cause your recombinant DNA to easily enter the cell, okay, through the pores that you created in step 1. And then after you have given the heat shock for 2 minutes, you immediately put them back, put this mixture back on ice. So after doing your selectable marker analysis, like you have to check, right, whether cell has taken up the foreign DNA or not. When you do that using your selectable markers, you will see whether or not that cell is transformed or not. Okay. Now there are other methods to introduce foreign DNA into host cells other than the steps that I just described. One of these methods includes the microinjection, where the recombinant DNA is directly injected through fine needle into the nucleus of the animal cell. So this is usually done for animal cells or the mammalian cells. There's another method called using gene gun or biolistics. So here what happens, you coat microparticles of gold and tungsten with your recombinant DNA. After that, you bombard these particles, microparticles at a very high speed on the plant cells. So this gene gun method is used for transforming plant cells. Microinjection method is used for transforming animal cells, and the use of calcium chloride or the calcium phosphate, and then using ice, and then you giving a heat shock. So this is how you can make a bacterial cell competent. Another method that has been used to transform host cells is by using disarmed pathogens. So we have just seen that Agrobacterium tumefaciens can be disarmed by removing its tDNA. Similarly, retroviruses can be disarmed by removing its cancer-causing genes. And so that once it is disarmed, they will be allowed to infect the plant or animal cell, and while it does so, it will introduce your gene of interest within the plant or animal cell, that is your host cell. Now there are certain steps. Till now you have understood what are the tools that are used in recombinant DNA technology. Like you have to use restriction enzymes, then look for suitable vectors, cloning vectors, in which you have to see that there must be suitable cloning sites. Other than that, you need DNA polymerases and then DNA ligases. Correct? You need a particular host type, and you must be aware of what kind of gene you want to introduce. That is the gene of interest. So these are the tools. Uh, Now we have to understand what are the steps involved in actually making a genetically modified organism, and then ultimately getting a desired product from that particular organism or a cell. Okay, so what are these steps? First step is you need to isolate the DNA. So you have to know that say from cell type A, you want you want to get a gene, and that gene is your gene of interest. So for doing that, you have to first isolate the complete genome from that cell type A. So this is step one. Step two is fragmentation of DNA by restriction enzymes. Okay, so you have to digest or make fragments of your isolated DNA as well as your vector DNA by suitable type of restriction enzyme. Then you need to isolate your desired DNA fragments. After you have done that, you can now ligate your DNA fragment into the vector DNA to create your recombinant DNA. Now these star marks, here also a star mark there, refers to an optional step, that is PCR amplification. Okay, what is it? I'll come to it later. Okay. Now the fifth step is after you have made your recombinant DNA. So here recombinant DNA is ready till here. You will now insert it or transfer it into your host. After doing that, your host cells will be selected, correct? With selected using selectable markers, means you have to select out the transformed from non-transformants. Then you have to select out the recombinants from non-recombinants. So after you have done that, you will culture only the recombinant host cells in a media at a large scale. Why? because you want to get the protein which your foreign piece of DNA or the gene of interest you selected. Okay, so you want to get that particular protein. So that is your last step. That is extraction of the desired product. That is desired protein. Now let's understand each of these steps one by one. So how would you isolate a DNA? Isolate DNA or isolate the complete genome from a cell. For doing that, there is a standard protocol for it. First step is you need to break down the membranes, the cell membrane, the nuclear membrane of the cell. So this will ultimately release all the macromolecules which are present inside the cell, like DNA, RNA, proteins, carbohydrates, lipids, etc. Okay. So how would you lysis? How would you do the breakdown of the membranes or lysis of cell? You will do that by using several types of enzymes that are available. Like for bacterial cell, people use lysozymes. For plant cells, people use cellulase to break down to cellulose cell wall. Then for fungus cells, people use chitinase to break down the thick chitin coating. Okay. So after you have done breaking down the cell membranes and all the cellular contents is now out in the solution, you have to digest the unwanted macromolecules so that ultimately you are left behind with the DNA. So what are the unwanted macromolecules? These are the RNA, proteins, lipids. So RNA can be digested using ribonucleases, proteins can be digested using proteases, lipids can be digested using lipases. Okay. Now, after this, you are only left with the DNA molecule. So from the reaction mixture, you have to easily precipitate out. Okay, that is precipitation step you will do. How will you precipitate out your DNA? You will do that by adding chilled ethanol. And this ethanol should be very pure. Okay, almost 99% pure ethanol is added and it should be chilled prior. Okay. And after you add um, ethanol, chilled ethanol, the DNA in its pure form will be obtained. So it would look something like this. And since the DNA content is quite large, it is visible to naked eyes and it looks like thread, okay, white colored thread. So this is how your isolated DNA will look like ultimately. Now this complete genome, which is consisting of maybe thousands of base pairs, maybe 10,000 or even lakhs of base pairs, you have to digest, you have to make it into fragments so that ultimately you can select out your gene of interest. Means, and your gene of interest is a part of the complete genome, correct? So for doing that, you will need to use restriction endonucleases. Okay. So how we'll do that? You will take your purified DNA, right? Which is, let's call it as source DNA. Okay. And you will incubate it with the particular type of restriction enzyme. Okay, say EcoR1. Now, you also need to incubate your vector DNA, like a plasmid vector, like PBR322. You also need to incubate your plasmid vector with the same restriction enzyme, EcoR1. Okay, plus, but they should be done in separate vessel. Like in one vial, you have your source DNA plus EcoR1. Another vial, there is plasmid DNA plus EcoR1. And the conditions of conditions in which these vials are kept, they should be at optimal condition so that the enzyme, restriction enzyme, can work at its highest activity. So once it is doing its activity, it will make fragments, DNA fragments. Now these DNA fragments need to be separated. Okay. So for separation, you have already learned the method agarose gel electrophoresis, where the DNA fragments are separated based on their sizes on a porous gel matrix, that is on a porous agarose gel matrix, where under applied electric field, the DNA fragments, they migrate from the negative end to positive end. Okay, so because of which you will get bands corresponding to different lengths of DNA fragments. So if you know what is the length of your gene of interest, you can then cut out that particular band from the agarose gel and do elution and get your purified gene of interest. Once you have got your gene of interest, you will do the fourth step, that is ligation. Ligation with your plasmid vector. Now your plasmid vector is also cut is also cut with the same restriction enzyme like EcoR1. So because of which there will be complementary sticky ends on both your gene of interest and your plasmid vector. So using these sticky ends, you will add, DNA ligase. But the DNA ligase will act on the sticky ends and form phosphodiester bonds as the sugar phosphate backbone, ultimately making a continuous DNA molecule where one segment is of the vector DNA. Another segment is a very small segment is of the gene of interest. So this is what you are already familiar is your recombinant DNA. Now let's talk about an optional step, that is a PCR amplification step. Now what does PCR stands for? PCR stands for polymerase chain reaction. Okay. So this is a reaction which helps in making multiple copies of a DNA molecule, okay, or a DNA fragment. Now this DNA fragment can be anything. It can be your gene of interest, small gene of interest, or it can be your vector DNA, or it can be your complete recombinant DNA, or it can be complete genome. Okay, so any DNA fragment or any DNA molecule can be subjected to this PCR reaction in order to make multiple copies, and these copies are all identical to each other. And this reaction happens in vitro, that is in a tube. In a tube it happens. That is why the name amplification is given, because here amplify refers to making of multiple identical clones. Now how to make a PCR reaction mixture? Okay, how to make a PCR reaction mixture, what all things you need to add into this mixture. So the volumes that we are going to use for PCR mixture, they are very small, like in microliters, remember. Okay, it's a very uh, sophisticated and small amount reaction we are talking about in microliters. So people have to practice for years in order to perfect this technique. Okay, but nowadays many resources are available, so uh, by simple understanding anyone can do, but yes, practice is needed. So what are things you need to mix? You need to mix obviously sterile water. You need to use small volume of sterile water because this reaction will take place in a aqueous environment. Next you will add a buffer containing salt, magnesium chloride, because this magnesium ion is important for the activity of your polymerase, DNA polymerase. Then you need to add your template DNA, means the DNA molecule which you need amplified, like your gene of interest or vector DNA or recombinant DNA. Then you need to add primers. So what are primers? Primers are nothing but small chemically synthesized oligonucleotides, okay, that are complementary to the regions of some region of a DNA molecule. Okay, now these primers can be either oriented in the forward direction or in reverse direction. You will understand the significance of each of these components in the PCR reaction in the next slide. Next, you need to add DNA polymerase, and here the DNA polymerase used is called Taq polymerase. Why Taq? Taq actually comes from the name of the bacterium, Thermus aquaticus. Now, this bacterium actually grows in a very hot environments like hot streams. Okay, you must have seen right from the ground, hot streams are coming like a fawara, like a fountain. So where the temperatures are above 100 degrees Celsius, so these bacterium are able to live there. So from this particular bacterium, people have isolated this DNA polymerase, which is now called Taq polymerase. A speciality of this DNA polymerase is that it can show activity at a very high temperature, like close to 100 degrees Celsius, without undergoing any degradation itself. Because if you are aware that enzymes are very much, since they are proteins, so they are very much susceptible to degradation, and especially thermal degradation. So by using Taq polymerase, people have been able to, overcome this particular problem, that it will not undergo degradation at very high temperature. Now, why high temperatures are used in PCR, I'll come to bit later. Then a sixth component that is unique to mix is DNTPs, that is deoxynucleotide triphosphates, right? That is ATGC, AGCTG. Okay, so these are things you need to mix to make a PCR reaction mixture. Now let's discuss what are the steps in PCR reaction. So PCR reaction is consists of cycles. It does it. So in one cycle of PCR, one DNA molecule gets amplified to create two more DNA fragments. Okay, or two more DNA molecules. And similarly, in exponential manner, the DNA molecules get amplified. So and within each PCR reaction, there are these three steps that happens repeatedly in each cycle. First step is denaturation. So in this figure, you will understand that, say, this is your double-stranded DNA molecule. In denaturation, what you do is you supply a very high amount of temperature, that is close to 94 degrees Celsius. Now, this heat, this high temperature is going to cause break in the hydrogen bonds that is holding together these two double-stranded DNA. So under this heat, the double-stranded DNA has now become two single-stranded molecules. Okay, so now these are two single-stranded DNAs. This is the denaturation step. So as you can see, the denaturation step is happening at a very high temperature, that is close to 94 degrees Celsius. So in the PCR mixture where you have added your DNA polymerase, now you must be able to understand why we are using a particular DNA polymerase like Taq polymerase in this PCR reaction mixture, because Taq polymerase can sustain this very high temperature, 94 degrees Celsius temperature. Okay, that is why Taq polymerase is used in PCR reaction, not any other type of DNA polymerase. Now, after the first step, where from double-stranded DNA molecule, you are getting two single-stranded DNAs with DNA strands, two single-stranded DNAs. You will do the second step. Second step will happen. That is annealing. Now, what is annealing step? Here, what happens? Remember, we talked about primers, which are synthetically made small oligonucleotides. Now, these primers actually get added in the forward direction and in reverse direction. How? There at these sequences, the sequence in the primer is actually complementary to a very small sequence, complementary to a very small sequence of both the strands. So this is this sequence which is going to cause the synthesis of the new DNA strands in the 5' to 3' direction, that is in forward direction, is called the forward primer. Whereas this particular primer, which is going to uh, cause DNA synthesis in the reverse, matlab, from 3 prime to 5 prime in this direction is the reverse primer. Okay, so these are the two types of primers, and these primers are usually very short, like only 15 to 20 base pairs. So while making these primers, you have to look into the DNA sequence of your template DNA or the DNA molecule that you have given for PCR amplification. So once these primers have complementary binded with the small regions of these two double-stranded, these two single strands, you then comes the step extension. Now, our extension refers to the synthesis step, that is the polymerization reaction step, where due to the action of DNA polymerase, here Taq polymerase, due to the action of Taq polymerase, deoxynucleotides are added at the 3' ends of the primers. Now, as it is doing that, a complete DNA strand is synthesized in both forward and reverse direction. So at the end of extension step, what you will get? You will get two new DNA molecules, correct, from one DNA molecule. So this is the product of one PCR cycle. In the next PCR cycle, what will happen? From two, four molecules will be made. Then from four, eight molecules will be made. In this way, exponentially, the number of molecules is increasing. So typically, people run PCR cycles for 25 to 35 cycles. So if suppose you have given 30 cycles of PCR, then you can assume that at the end of the PCR amplification, you will get around 1 billion copies of your DNA molecule, okay, of your template DNA. So that is quite a lot, and that is enough for subsequent steps. I think that will be enough. If you want more, then you can set for even more number of cycles. Now, once like this is an optional step, okay, so you can do PCR amplification or not. People generally do it in order to make stocks of their gene of interest or recombinant DNA, etc. So once you have done it, you will then transfer your recombinant DNA into a host cell. So you already know the techniques, how to do that. You can make a bacterial cell competent, okay, using calcium salts, right? Calcium phosphate, and then heat shock, and then exposed to ice. Then you can use microinjection technique or gene gun technique, or you can use a disarmed pathogen. So all these techniques are available, which will help you to transfer a recombinant DNA into a host cell of your choice. So now the recombinant transformed cells, host cells, they must be selected, correct, from the non-recombinant, non-transformed cells. How will do that? You will do that using selectable markers. So selectable markers that are available to us are the antibiotic resistance gene and the beta-galactosidase gene, whose working you are already familiar with, which I've discussed previously in this lecture only. Now the last step, second last step is that once you have obtained your recombinant cells, you need to culture them, okay? You need to grow them in a suitable nutrient media so that they express your gene of interest and secrete the resulting protein, and that protein is your product of the entire steps that you have done so far. So people do it at a large industrial scale. But before moving to industrial scale, one needs to optimize those conditions at a lab scale. So, cloning vectors, they are used for making multiple copies of a gene of interest within the host cells. But when one wants to express their gene of interest, then that is done using expression vectors. Now, expression vectors you will learn more about in your higher classes, like higher studies, if you are doing in this area. There you will understand more about expression vectors. But for now, know the fact that expression vectors are used for expressing your gene of interest inside a host cell, okay, inside a bacterial cell, say, or a plant cell or animal cell. Now, when these gene of interest is expressed in the recombinant cells, means your bacterial cell or plant cell, then the protein will be called now recombinant protein. It will not be a normal protein that you have just taken from a native cell. It will now be called a recombinant protein. Okay. So what steps that you need to do to make your recombinant protein? First, you need to culture your recombinant cells in suitable culture medium. Then you need to allow these cells to secrete the recombinant protein in the culture media. Now note, it may happen that the cells are not secreting outside. It may be synthesizing this protein which is present within the cell membrane. So an additional step of breaking down the cell membrane and releasing the cell content outside, and then do purification, that additional steps gets added. But for the simplicity's sake, we'll assume that the host cell that you have chosen for transferring your recombinant DNA is going to, secrete the recombinant protein in the culture medium. So once it does it, you will be able to purify. You will have to purify out or separate out your recombinant protein from the culture media contents. So ultimately, you will get your recombinant protein. So remember, all these steps needs to be optimized at the lab scale, and then only you will be able to move to industrial scale, where the volumes of reaction, etc., will increase. So in industries, people make recombinant proteins using something called bioreactors. Now, what are bioreactors? Bioreactors are nothing but huge vessels, okay, huge vessels, which are of volumes 100 to 1000 liters, okay. And these bioreactors can operate in two modes, batch mode or continuous mode. Now, what happens in batch mode is, at the start of the reaction, you add all the contents like the raw materials, cells, recombinant cells, um, any other thing that the cell needs to grow, okay. You add at the beginning, and you allow, say, um, to, the cells to act on the substrate and make the product, that is your recombinant protein, say for 2 hours, 3 hours, or whatever time period it needs. And then at the end only, you take out all the content, okay, from the reactor, that is the culture media containing your recombinant protein, and subsequently you will purify your recombinant protein. This is the batch mode. Whereas in continuous mode, what happens, you are continuously adding fresh medium and fresh substrate from one end of the reactor, okay, and from the other end you are taking out the culture medium containing the product. So because of this, what happens? There is a continuous circulation of fresh medium for the cell and removal of waste products, as well as you are continuously removing out the recombinant protein, okay, from the cells. So usually what happens, cells are often sensitive to the concentration of the proteins. So in those cases, a continuous mode of operation would be beneficial. Now, bioreactors needs to maintain optimal conditions for allowing proper growth of the recombinant cells. Okay, so that these cells can convert the raw material, the substrate, into useful proteins like enzymes or antibiotics, etc. So, what kind of optimal growth conditions cell needs? It needs a proper amount of oxygen, proper concentration of oxygen. Okay, if it is an aerobic type of bacterial cell, there are anaerobic cells as well, like yeast, etc. Then you need to give proper temperature for its growth. The pH should be optimal. The salt concentration or the iron concentration should be optimal. Cells should be provided with required amount of vitamins, etc. And obviously the substrate which the cell will consume, okay, and use it for its growth, as well as use it for the synthesis of the recombinant protein. So these are two commonly used bioreactors. Okay, one is stirred tank, another is sparged stirred tank. Now, in both these images, you can see that bioreactors are nothing but a very large vessel. Okay, and within the vessel there is a huge rod. Okay, where agitators are connected. Okay, what do you mean by agitators? I mean blades, okay, or propellers are connected, which are moving in a and which are rotating continuously in clockwise or in anti-clockwise direction. So these blades are allowing continuous circulation of the uh, contents of the bioreactor. So as well as it will allow easy reach of the nutrients to the cells. Oxygen will be uniform, amount of oxygen will be uniform within the bioreactor. As well as you can see, there are control systems like which will check which will check frequently the pH of the uh, contents of the bioreactor. Or there are also temperature control systems. Okay, then there is something called foam breakers, which will stop excess amount of foam leaving the reactor. It will scoop out the foam and then remove it from the reactor from one port. And there is continuous flow of sterile air. Okay, means continuous flow of fresh air containing oxygen. So these are the components of the stirred tank. How sparge tank is different? Here what is happening, there is a section through which oxygen is bubbled in, okay, within the media or the cultured broth, within the content of the bioreactor. Now the aim is same as in stir tank reactor. That is, we want we want continuous and uniform circulation of nutrients and the oxygen, okay, around the uh, entire volume of the bioreactor. Means each cell should be able to get fresh media and fresh oxygen. So for that purpose only, uh, gases, any gas like oxygen, etc., is uh, forcefully pushed into the content of the bioreactor. Here, agitators are there. Here, gas is spurged. This is called spurging actually. Now, bioreactor components that I've already mentioned, but I'll repeat here, consist of agitator systems, that is the propellers which are continuously rotating. Then there is oxygen delivery system, okay, that supplies fresh oxygen, and foam breakers or foam control systems, temperature control systems that monitors the temperature of the broth, culture broth, then pH, that monitors the pH of the broth, as well as there are small ports called sampling ports from which any person, any technical person can go and take out few volumes of the culture broth at regular intervals and check how far the reaction is along. Okay. Now, last step after this is you just need to take out the culture broth, okay, and do what is called downstream processing. So downstream processing includes separation of the unnecessary, unwanted components of the culture broth from the required protein, desired protein. First, you need to separate. Then you will need to purify further the obtained protein, recombinant protein. It should be in most pure form. More and more you can make it pure, more valuable it is. It will have its commercial value. Okay. So these two steps, separation and purification, these two steps under the broad umbrella of downstream processing. Now, after downstream processing, the highly purified form of recombinant protein can be now mixed with suitable type of preservatives. That is, going to increase the shelf life of your protein. Now you can actually take this product to clinical trials. Well, it will be tested on humans and check whether or not it is safe for use or not. And ultimately, if this product passes the clinical trial test, means if it proves that it is safe for human consumption or human use, it will be approved by concerned regulatory authority for sale in market. Okay, whoever is the manufacturer, any big company, they will be able to sell this product in the market, and people can, common people like you and me, we can go and buy. So this is how biotechnology products like enzymes, proteins, antibiotics, hormones, etc., which are available as drugs, etc., these are made. Okay. In the next chapter, we'll discuss more in detail through few examples on how various kinds of drugs or hormones, etc., or a different type of genetically modified crops, how they are made through or using these steps of recombinant DNA technology that we have discussed so far. So thank you for joining today's lecture. So now I'll end this session. Thank you.